My screen is shared with you, right?
Yes, it's shared.
Okay, great. Yeah, thank you so much. Good afternoon, everyone. My name is Mohammad Soleimani. I'm a member of, as a PhD student, I'm a member of Laboratory of Physical Chemistry and Center for Multi-Scale Electron Microscopy at Eindhoven University of Technology. And during my PhD project, we work on a living organism called Diatom, and we try
Leuk
to.
verhaal trouwens hoor. Ja, dus best een leuk verhaal heeft hij.
Is there something wrong?
Everything is fine.
Or
Somebody
if-
was
okay.
unmuted by accident.
Yes, it's all right. Yeah. And in our research, we try to find out the effect of environmental condition like the presence of aluminium ion on the growth rate and different parameter like morphological, chemical, and mechanical properties of silica in diatom. Diatom is a unicellular photosynthetic algae which can live everywhere that we have water, from ocean to lake and river. Even some of them live in soil. We have more than 10,000 different diatom species, and every diatom has its own unique shape and structures. And regarding the size of diatom, it's most of them between 2 to 200 nanometer, the diameter and the length of this scale. Some of them are at the range of millimeter, but most of them are at the between 2 to 200 micrometer. They are responsible for carbon fixation. 20 to 25% of the carbon dioxide assimilation in general is done by diatoms, which is equivalent to all rainforest. And that sense, they have a huge impact in our lives for producing oxygen. They are at the bottom of marine food web, which means they produce food for other organisms. And as I said, every diatom has its own unique shape and structure. The most interesting aspect of diatom is their silica exoskeleton, which is made based on silicification or biomineralization of orthosilicic acid from the environment. Generally speaking, we have two classes of diatom, centric diatom and penic diatoms. And you can see each diatom has its own unique shape and pattern with micro and nanopore structures. Usually there is an organic layer around the silica structure of diatom. By chemical treatment or thermal treatment and removing this organic layer, really highly ordered micro and nanopore structure of the exoskeleton of the frustule can be seen, which is known as frustule. And there are a lot of functions for this frustule. For example, they act as a protection of the species against other organisms. The way that diatom growth is based on cell division. During the cell cycle, one mother cell gradually divided to two daughter cells, and this cycle goes on and on as long as there are still enough nutrition in the environment for the species to take up and to grow. The structure of diatom is like a petri dish. We have two lids, which are known as valve. The top one is called epitheca, and the lower one called hypotheca. And these two valves are overlapping each other by a number of Gradle bands. The way the diatom makes this silica structure in biosilicification, they take up orthosilicic acid from the environment, is a soluble form of silica, and with a specific protein known as silicic acid transporter protein, they transport this silicic acid from the environment to inside of the cell. Usually there is an organic lipid. There is a lipid layer around the silica structure, and this protein helps the organism to take up silicic acid, which is the main ingredient for building the silica structure. And after taking up the silicic acid, the organism transferred the silicic acid to a specific compartment known as silica deposition vesicle. Inside the silica deposition vesicle is a membrane, is another membrane. Inside the silica deposition vesicle, the condensation and the polymerization of the silica takes place, and finally form. And then, as you can see here, after completion of the silica structure, based on the exocytose mechanism, the silica which is formed inside of the cell moves toward the outside of the cell, and then a new griddle band is formed, and this process repeated as long as we have silicic acid in the environment. In our research, we want to feed this organism with aluminium and to see what's the effect of aluminium when this organism takes up aluminium and build the silica. For this, we need a climate cabinet. As I said, they are photosynthetic, they need light. And with climate cabinet, with a control light and light intensity and temperature, we grow this organism. Usually in the cultures that we grow them, they need a lot of chemicals. One of the essential one is silicic acid. As long as silicic acid is present here in the culture, the diatom can take up silicic acid and creates a new silica structure, valve, and griddle bands. We can do the synchronization, which means all of the diatoms are growing at the same age, and after synchronization, we can end the synchronization by feeding the organism by silicic acid, and this time, because we wanted to incorporate aluminium in the next generation and the new generation of the silica, which is formed, we feed them with aluminium to incorporate aluminium alongside silica into the silica structure, alongside silicic acid into the silica structure of the frustule. Usually under light microscope, you see something like this, yellow brownish color of diatoms, which is not really interesting because there are a lot of organelles inside of the diatom, and as I said, there is an organic lipid, organic casing around the frustule outside. And in our research, we want to see the effect of aluminium on different properties, like mechanical, morphological, and chemistry of the silica when we feed the diatom with aluminium. In our lab, we work on 2 diatoms. We grow them in a climate cabinet. We feed them with all of the nutrition that they need, and we feed them with aluminium as well. Here you can see an intact frustule of diatom, the 2 lid, the bigger one and the lower one, and the griddle bands which are connecting these 2 valves to each other. Here you can see the griddle bands which are overlapping each other. An isolated valve is here, which contains a lot of features like raphe, central nodule, and the porous area. In the porous area of the frustule, we have a different feature which we can track some with image analysis throughout the generation, and to see the effect of aluminium on these features as well. We have another diatom species, it's a centric diatom. In this case, again, we have 2 valves at the top and the bottom, and the griddle bands which are connecting these 2 valves. Isolated valve, the valve contains a lot of features like a branch and cross connection and small pores. It's really complicated, the structures from outside, but from inside of the valve, there is no any pattern, and the structure like outside of it, and it's more or less smooth. But the small pore is clearly visible here, and over here you can see an isolated valve of these organisms. After feeding this organism with aluminium and other chemical, we wanted to see whether aluminium is inside the silica or not, and for that we did CM EDX analysis mapping actually. And for both cases, for both diatoms, we saw aluminium alongside oxygen and silicon in the valve, and in this case, even in the griddle bands, we see the homogeneous distribution of aluminium alongside silicon and oxygen, which is an indication of presence of aluminium in our sample. And then we did study the state silicon NMR to see the coordination environment of aluminium in the silica of diatoms. For example, for this one, we work on different concentration, and in both cases, we have 4 and 6 coordinated aluminium, and in this case, most of the aluminium was 6 coordinated. I have to say that 4 coordinated aluminium is the aluminium which is inside of the silica framework, but 6 coordinated is the aluminium species which usually is on the surface or is not connected inside the silicon network. For this species, we track the different morphological feature to see the effect of aluminium on the different feature, the different morphological parameter of this organism. We found out that the length, the width, and the area of the valve of this organism doesn't change. They don't change by changing the concentration of aluminium from 0 to 2 micromolar. 2 micromolar is the highest concentration of aluminium that we use for growing these organisms. And all of this concentration found in nature, and we try to be really close to the concentration of aluminium which we can find in different region in nature. But closer look to the valve, we found there are some differences in terms of sample with and sample without and sample with aluminium. From A to D, you can see there's a difference in terms of pore size. The A is without aluminium, and D is the sample with highest concentration of aluminium, 2 micromolar. We found that the overall features of the pore they don't change that much by changing the concentration of aluminium. For example, the distance between the pore from 2 array or even in 1 array, the distance between the pore they don't change that much, but the diameter of the pore, by increasing the concentration of aluminium from 0 to 2 micromolar, you can see the pore size is decreasing, and it's more or less is linear behavior. And the next step, we wanted to see the effect of aluminium on biosilicification of the sample and how they grow actually in the presence of aluminium. We measure the cell density over time of the when we feed the diatom. We at a different time we collect them and we measure the cell density, and we found out by C0. I have to say that C0 is the sample without aluminium, and C3 was the highest concentration of aluminium. And we found that a negative effect of aluminium, for example, in this case, the culture with highest concentration of aluminium has a lower growth rate in comparison to the sample with high concentration of without aluminium. Actually, aluminium has a negative effect on the growth rate of the species. But at the same time, we measure the silicic acid uptake, which is taken up by diatom in the culture. Surprisingly, we didn't see a huge difference from one culture to another, which means the diatoms, they grow in the presence of aluminium. We have fewer number of diatom, but the amount of silicic acid that they take up is more. And for that, we did TGA, and we found out that we have more inorganic content in the sample with aluminium in comparison to the sample without aluminium. And also based on the cell density measurement and silicic acid uptake, we determined the silica content in the sample, and the sample with highest concentration of aluminium, we have around 60% more silica in comparison to the sample with without aluminium actually. And it was really surprising because the only difference that we found was just the pore size of the valve, and there should be something else is changing in the presence of aluminium. For that, we did TM image analysis to measure the thickness of the valve. We measured the thickness of different regions, for example, the porous area of the valve. And as you can see here, clearly from 0 to 2 micromolar aluminium, there is a clear difference in terms of the thickness of the valve. Actually, we measured the different region, for example, the raphe and even the whole valve, and it was a linear increase in terms of the thickness of the silica in this species. Another part of the work was to see the internal structure of this organism when we grow them in the presence of aluminium. We did focus IMM slice and view. I'm sure most of you know what the process is, but it's like a bar of a cheese and a knife. You cut the first part and you take the image and you go through all of the sample and you collect the image and you can reconstruct the structure. And in this case, the samples which we grow them in the presence of aluminium, we found some kind of high contrast particles at a different part of the sample. And even with the sample over here, you can see the diatom, which the other cell have already been separated and they're ready to completely go to outside of the cell. Even in this case, we saw that there are some kind of particles, because it's backscattering detector, these images. We saw these particles, which we didn't see them when we don't feed the diatom with aluminium. And we did the EDAX mapping on this sample to see the elemental composition of these particles. I found out that they have a lot of phosphorus, aluminium, and oxygen. And we know based on the literature that aluminium phosphate particle is like forming aluminium phosphate particle inside living organism, including diatom, is a kind of a detoxification mechanism to deal with aluminium, because aluminium is not doesn't have any biological function. And even in our case, we saw that it decreased the growth rate of the species as well. But the next step of our work, we want to measure the mechanical behavior of our samples with and without aluminium. For that, we use a micromanipulator inside the microscope. We can mount them inside the SEM chamber. This micromanipulator can move in 3 axes with high positional accuracy of about 10 nanometer. And you can mount a different probe on the micromanipulator. You can mount a micro gripper. In this case, you can see 2 arms of a micro gripper, and I am collecting one of the valve of one of our species to move it from one place to another to do further characterization. You can mount a sharp tungsten tip to create a pattern on the surface with a high accuracy. You can even collect some small particle, in this case 300 nanometer silica particles, to move them from one place to another to create a kind of a nanostructure. But the most interesting part of micromanipulator for us is to mount a force sensor on it and measure the mechanical properties of our samples.
5 more minutes.
Yes. I have to skip this one. Yes, for our sample, for this sample, we saw that we have a sample with aluminium. We have a thicker valve, and the pore size is smaller. And we did some preliminary study to see the effect of aluminium on at least the force necessary to break the valve. And in this case, we saw a clear difference in the sample with and without aluminium. It reached a point of the fracture. In this case, you can see it here. We saw a difference in the sample with and without aluminium. And as you can see, that the fracture took place throughout the central nodule and the porous area. And this one is an indication of the thickness and the pore size as well. We have a different species as well, the TP1, which is a centric diatom. We can do the mechanical study on it in a different way, like doing the compression on entire sample or indentation when it's lying down on griddle bands to measure the mechanical properties of it. Or even you can do it when it's standing on a valve. In all cases, you can see the elastic behavior of this species is completely coming back to its original shape in this case, or even when you are applying the force from the valve, you can see the elastic behavior of this one. But the problem with that, we have different kind of a behavior, like bulging, buckling, and the modeling of this organism later on will be really difficult. And another difficulty of this one is the pattern of the valve. It's really complicated to be modeled, and we need to simplify this structure. And first we need, for mechanical study of this species, first we need to preserve the structure. By air drying, usually the whole structure collapses because the thickness of the girdle bands is less than 30 nanometer. And first we need to preserve the structure by critical point drying. And then we have to remove the valve to make it as simple as possible, something like this, to which we can easily do the mechanical study on it, and later on the modeling of hollow open-ended cylinder will be really easy. That's an ongoing research that we are working on it nowadays. And finally, as conclusion of our work, we by incorporation of aluminium in our sample, we changed the pore size and the thickness, and by solid state aluminium NMR, we found that we have 4 and 6 coordinated aluminium in our both cases. And nowadays we are doing a lot of mechanical study inside a microscope, and meanwhile we are doing solid state silicon NMR to see the effect of aluminium on cross-linking of silica. With that, I would like to thank my supervisor, Rolf van Bentem, and my daily supervisor, Heiner Friedrich, and also my colleagues for all of their help and support. And of course, thank you for your attention.